Oké dan. So hello, in this lecture I want to tell you about how the sequencing with the use of Illumina sequencing platform really works. So how to start off with your RNA and how to process it to finally get your raw sequencing data. So starting off, of course, as this is RNA sequencing, we'll be starting off with RNA. So RNA usually can be used for RNA sequencing is just total RNA. It depends a bit on the kit you're using, but usually the thing to go off with is just using total RNA. So this does not only include your messenger RNAs, mRNA, but also stuff like tRNAs, ribosomal RNAs, and non-polyodylated RNAs. So in this picture, you can see RNAs of different quality. So you want to make sure that the quality of your RNA is good. And one thing to use, especially when using total RNA, is you can look at ribosomal RNAs. So these ribosomal RNAs, is the 28S and the 18S, are visible as two strong bands, as they are highly abundant in the samples of most organisms. And the intensity of the bands, it can tell you something about the quality. So if these are breaking down, they will also disappear. So which you can see in this picture. On the left, you see RNAs of poor quality. What you can basically see is the top band, 28S band, is almost fully degraded. And you can see a grayish smear between 500 to 2000 bases. And if you then go to the panels to the right, you can see a higher quality with the ones in the right, the three samples, which have a really dark 28S band and also a very clear 18S band. And in the bottom, between 1500, it is almost white. So mRNA, where can you find it in these samples? So as mRNA is present in a multitude of different lengths, so you do not see them as bands or anything. They will just be in the background as a smear. And also the mRNAs are only a few percent of the total sample, so you just cannot really see them. So in this case, when using RNA, it is very handy to use the ribosomal bands to quantify the samples. Right, of course, sequencing cannot be done with RNA. Sequencing is more or less like a PCR reaction with DNA polymerases, and you need to have DNA as template. So before starting with the sequencing, your RNA, your samples need to be prepared to end up with DNA or cDNA, copies of your RNA, with the proper adapters and stuff around it, which actually fit to the flow cells. So for that, the samples need to be prepared, which is also called the library preparation, the library prep. And this is usually done with a kit. So here are some examples from different companies who have kits, which you can just buy, and then you can just go step by step through the protocols. And then your samples will be ready for the sequencing. So how does this actually work, this library preparation? So in this case, I'll just show you one example of a basic way of preparing a library. There's more ways to do this, but what you usually do is, so starting off with your RNA, and what you typically want to sequence is your mRNA, the messengers, which have a poly-A tail in the end. So to start off with making cDNA, it is usually used an oligo-DT, which is a primer, as you can see down here, which contains a stretch of T's, which binds to the poly-A tail. And behind that, you will find an adapter, which is basically a sequence, 20-25 bases, with a certain sequence that is necessary for downstream steps that will be shown later. So after binding of the primer, a DNA polymerase, so an RNA-dependent DNA polymerase, an RT polymerase, will make a copy of the RNA, of course, in reverse complemented orientation. And so you have your first strand cDNA. And the next step is that the RNA will be degraded. So with an RNA degrading enzyme, the RNA is completely removed, leaving a single strand of cDNA with poly-A tail in the end, or an oligo-DT, no, poly-T tail in this case, and the adapter. So the second strand that needs to be made is then done with so-called random primer. So a primer is added with on this side 6 to 8 bases, different sequence. So every primer has a different sequence, which is basically a random sequencing of 6 to 8 bases. So every sequence will be in there, so it can bind to every transcript in your sample. Next, again, DNA polymerase will elongate the primer and make the second strand of your cDNA. So of course you have to take in account that if you have multiple transcripts from the same gene, that every time a different random primer will bind, and this will lead to different fragments, so that you get different sizes of your double-stranded DNA. So even though they all come from the same gene, you can still get different fragments, right? So the next step is to amplify this and also to add more sequence and the indexes. So starting off with your double-stranded cDNA, it will be amplified with a forward primer, which has the green parts, which can bind to the cDNA. And then next you see the purple parts, which are typically 6 bases, sometimes 8 bases, but they contain the so-called barcode. So this, for each of your samples, you will add a different primer with a different barcode. And after sequencing, you can again find back from which sample the RNA is coming. And in black, there's more adaptive sequencing. So the whole thing is about 50 to 60 bases with in the middle a barcode, which is specific for your sample. On the other side, so usually, typically with RNA sequencing, the number of samples is not that big. So this is not used. So the primer is usually the same, so they don't use different indexes for different samples, but just stick to one. But if you have a lot of samples, also for like single cell sequencing, where you have a few thousand different samples, then you will also vary this barcode. And you have 2 different barcodes on both sides. Anyway, these primers are used, they bind to the sample. And again, after a PCR, you get your final cDNA library preparation, which will be containing of in gray the copy of your RNA, including the polyA tail, and then on both sides an adapter. And of course, these adapters are different from each other. So after the library preparation, the samples will be mixed. So they checked for quantity and quality, and then mixed in equal amounts, so that you get the same number of sequences from all your samples. And the number of samples can vary a lot. If you want to read in depth, you will have a limited number. And if you just want to read a lot of samples for a relatively low number, then you can also mix lots of different samples. So how does the sequencing itself work? So we're starting now off with these libraries. And these libraries are denatured. So with the use of some heat, it is made single-stranded, and then they're added to a so-called flow cell. So for a Illumina flow cell, is about the size of a microscope glass. So it's about 7.5 centimeters long, 2 centimeters wide, and it contains 8 lanes, they're called, in which liquids are flown across. Hence the name of flow cell. Flown across, flowed across, flowed across, excuse me. On these lanes, you can find 2 types of oligos. In this case, indicated as the orange and the greens. And one of these 2 will be able to bind to this part, the blacks, and the other one will be able to bind to the orange on the other side. So looking here at the surface, where you have these 2 types of oligos, your single stranded CJ molecules can bind. And in the next step, this needs to be amplified again on the flow cell, which is done with a so-called cluster PCR, bridge PCR also known, in which so the first strand, of course, is just made in single. So the oligo that is bound to the flow cell is elongated. And then after denaturing, so this is again heated, the black strand in this case will be single-stranded again and washed away, and ending up is a single strand of cDNA, which is covalently bound to the flow cell. So that is the first strand. And next, a bridge PCR will be performed. So again, this single-stranded, the other adapter of your single-stranded molecule can bind to the next adapter or PCR primer, which is bound to the flow cell, which forms like a little bridge. It's named bridge PCR. And then this one is again elongated. And then after denaturation, there is two molecules bound. And this is again repeated and repeated and repeated until you get this cluster, which are all like identical copies of the original molecule this was attached. And as this is just the first molecules are just randomly applied to the flow cell, you will get these little clusters, which are all as copies of a single molecule. As we look at the surface of the flow cell. So next we get to the sequencing itself. So we have this DNA molecule again bound to the flow cell with the adapters, in this case indicated in pink and blue. And then if we zoom in, the sequencing primer is added, which is just a simple primer that fits to one of the adapters. And zooming in, something like this. So it binds to the adapters, just flanking the cDNA fragments. Next, DNTP are added. And these DNTPs contain a block. So a single DNTP nucleotide can be connected to the primer and elongated to the primer. But because of this block, it cannot continue and the next one cannot bind in this case. So next, these the samples are exposed with a laser, and then actually first the DNTPs are washed away that are not bound, and then next the samples are exposed to a laser, and each of the nucleotides has a label of a different color that will be shining up and giving a signal. In the next type of this cycle, the label and the block will be removed, and then the steps are repeated. So DNTPs are again added, a laser exposes the samples, the glass, the flow cell, and it is detected again which color is visualized, and in this way each of the bases are, every cycle, a single base is detected in repeated steps called cycles. And typically for a reaction like this, a run consists of 50 to 150 cycles, so giving a sequence of 50 to 150 bases. And optionally, of course, the run can be done from starting from the other adapter and then reading from the other side, which will lead to a so-called paired end reads, where you have two sequences from a single fragment. And of course, in this case, the techniques are not strong enough to really read a single base pair. So that's why we started off with these clusters, which are all the same molecule. So all these molecules will shine the same light, as they have the same sequence, and then you will, the whole spot, the whole cluster will light up in each cycle, and that will lead to something like this. In this case, this is a part of the flow cell in which the pictures of the different cycles are just visualized. You can see all the different colors for the different bases that are integrated. So looking again at a single fragment of the flow cell, where you can see different colors for the same cycle. So different color is a different base. In this case, the green is a T. You can see that this green is a T and this green is a T. And in the next cycle, for instance, you see that this one is blue now and still green. And the bases are added, and this is continued each of the cycles, as you can see. And sometimes when you have a lot of the same sequence, it still gives a clear sequence. So, and of course, as you can also see in this picture, that some of the spots, like the ones that are circled, are really clear, they're nicely separated, they give a strong, clear signal. So there's no doubt whether this is green and this is blue and this is red. So the sequence is clear, so a lot of high quality. But if you look like here on the side, you have a bit of a whitish one, and you have some yellows and with a bit of red in the bottom maybe. So In this case, of course, the sequence is not as clear. And because you do want to have like an indication how sure you can be of the sequence. So for each of these spots, they not only get a sequence, but they also get a quality value. Yeah. So this quality value is just makes use of characters. So just all the characters on your keyboard. And these characters have just a different value. Exclamation mark is a 0, a 2, a 3, and a 4. Plus is 10. And after all these characters, you get the numbers. And a question mark, for instance, is a 30. Then you get the capital letters up to 40. And this value, this number, is an indication for the quality of the read. So if a fret score, if this score is, if your base has a value, you know that the quality score is 10. So a probability of an incorrect base is 1 in 10. So your accuracy is 90%. 20 would be 1 in 100, 30, 1 in 1,000. And if you get to a 40, you have an accuracy of 99.99. And of course, these numbers are just in between these values. And again, this is all based on how clear the clusters, the spots can be read. If they are nicely separated, you give a good color, you get a high value. And if they are close together and a bit awkward to read, then the value is lower. You get a lower score. So sequencing is not done in a single read. So you get the sequencing where you have your primer bound to the sequencing primer site, which is in this case the green part, and it reads inwards. And this, as said earlier, can be done from 50 to 150 bases. After this 50 to 100 cycles, this part is removed by denaturing. Then another sequencing primer is added, which will do another 6 cycles of sequencing. And in this case, the barcode will be run. So the barcode or the index is run in a separate reaction from the sequencing of the cDNA itself. So what is the final output? This is a so-called FASTQ file, which contains all the reads, and all the reads are depicted in 4 lines. Within the first line contains the name, which is based on the position, so which lane it is from 1 to 8, and where more or less coordinates and where you can find it back with these numbers. And in the back, you can find the sequence of the barcode. So as you can see, these sequences are all coming from the same sample, and they all have the same barcode. So the second line is the sequence itself. The barcode contains the sequence itself, which is just the T, G's, A's, and C's, and sometimes you get an N. So as you can see in this experiment, you can see that everywhere, like where is my cursor? Yeah, so here there's an N in the 6th position here, but also here, and also here, and also here. So there must have been a glitch in the machine which made the 6th cycle give a bad read, leading to N's all over the place. So the third line is available for additional information. So usually it's just a plus, and sometimes it's plus and a repetition of the name. But they always say it is for additional information, but I have not been able to find any examples of this. But it is available. Fourth line, it contains the quality line. So this line has exactly the same length as the sequence. So for this T has a quality of A, this G has a quality of A, and etc., etc. So they have to be of the same length. If they are not, the programs using these files will run into another error. So here again, to have a look. So all the A's have high values, so they're in the 30s. So that's higher than 99.9% accuracy. And then, for instance, where you have an N, you have a very low value, a hashtag, which has a quality of 2. End of the slide. So for the sequencing, in the next presentation, I'll continue with how to work with these FASTQ files. Thank you.